All right, great. So thanks everyone for joining us, and thanks to the previous speakers. I'm Brian Delaney, Clinical Trials Liaison at RaDVaC, and I'll be speaking about understanding risk with vaccine trials, and specifically about challenge trials as a potentially safe way to get vaccines approved and deployed quickly where they're needed. So we've all seen these numbers. We're here at this summit because of an acute crisis, one that we think can be substantially mitigated with new ways of developing, thinking about, and deploying vaccines. But as all of us have stressed, we need to be thinking about how we can be prepared for the next pandemic, of course. What's preventing vaccines from being more widely deployed? Well, we've heard a lot of ideas about some of the bottlenecks. A huge part of it, as W.H.O has pointed out, is just the expense of trials. These items on the right here, these are a little bit more controversial, let's say, and we'll be speaking of these, and other speakers will be getting into this in more detail. And supplies are actually generally cheap, although sometimes, a bag or a test tube can suddenly run out and everything is halted. We've seen that during the pandemic. Our vaccine is actually not complex to produce, which is a huge advantage. But a large bottleneck by itself is this simple failure of rationality in assessing the risks of vaccines. So we like to illustrate this failure of rationality with the trolley problem, and lots of people like to use the trolley problem. And different components, as we saw in Ben Horvit's talk, can be analogized to different aspects of the problem. It's actually a very productive thought experiment. But I'll be focusing on this to understand how people tend to understand risk with vaccines. So again, to summarize, there's a runaway trolley that's heading down a track with 5 people immobilized. And you're standing next to a switch to move the trolley to a different track. If you don't switch the tracks, the trolley will very likely hit and kill all 5 people. But there's one person immobilized on the other track. And if you switch tracks, that one person will very likely die. Your inaction will result in more harm than your action. But if you take action, you will feel a more direct responsibility for whatever harm is done. How people react to this has been studied extensively as a matter of empirical moral psychology. By a bit of a majority, most people actually will throw the switch. And that makes sense. It's logical. We would hope that it would be everyone, or nearly everyone, who would do the math and draw the rational conclusion and throw the switch, but it's not. We seem to be concerned more with sins of commission than sins of omission. Now, this is a very complicated thought experiment, and there's a whole subfield, as you can imagine, having heard the previous talks and now mine, known as trolleyology, getting into various aspects of this. It's not clear what the general form of the trolley problem actually tells us. But reactions to the vaccine trolley problem are actually striking. And we'd like to use this in part because it really does highlight how people are simply not being rational about risk when it comes to vaccines. The trolley is the virus, and it is already hitting people who haven't been vaccinated. Now, people are worried about the ethics of deploying vaccines without years of testing. It seems to be that there is this over-concern, a hyper-concern with the risks of possible harm from vaccines and a seeming lack of concern with known serious harm from the pathogen. With the vaccine trolley problem, it seems that most people, the vast majority, would not throw the switch unless years of data are available, years during which millions of unvaccinated people in a pandemic are being killed. In practice, as we've seen, the situation is far worse. There isn't one person reflecting on this trolley problem, the vaccine trolley problem. There are many people in different regulatory agencies. And aside from in some countries, they have to reach a near consensus to actually pull the switch. And most or all of these people share this hyper concern with vaccine safety. Waiting for months of data before deploying vaccines widely might seem like a passive choice, just leaving the lever alone, to the extent that it involves following the inertia of recent convention of vaccine testing. But there's an unacknowledged active choice to allow countless people to go without protection. And as Ben Horvit noted, the choice not to rip up the tracks and lay them somewhere else. The recipients of placebo, but then also all the people who are in the trials are being exposed for many months potentially needlessly to this deadly pathogen. So one way in which the rollout is slowed is manifest in how often conventional vaccine studies are paused and authorizations are suspended. We all saw this headline and headlines like it. This was a study involving tens of thousands of people and was paused because of one unexplained illness. And the same thing with the AstraZeneca vaccine. Now, of course, we now know that the AstraZeneca vaccine is not perfectly safe, but no medical treatment is perfectly safe. And it's absurd to expect that this particular type of medical treatment, vaccines, should be perfectly safe. When it's non-deployment, clearly it is not. It's actually quite deadly. And during this pause, above all in the US, death tolls kept mounting. So we have a kind of separate trolley problem here, the vaccine trials trolley problem. One serious unexplained illness and trials have stopped, but the virus keeps rolling forward. This is a slide that Preston used, but I'm going to steal it because it really bears repeating. RaDVaC's perspective is that there's a clear asymmetry in risk assessment. People underestimate the proven known high risk of the virus. We just accept it because it's a serious virus, and serious viruses kill people. That's what they do. And people overestimate the typically very low risk of vaccines. We are deeply troubled by simply one person being harmed out of tens of thousands by a vaccine. In other words, I'm getting a note that my audio faded out. I hope it's okay now. Vaccine deployment has been irrational, and that is more of a general problem for trying to assess alternative ways of thinking about vaccines. And the proof of that irrationality couldn't be clearer. If we understood risk more clearly, we wouldn't simply more readily allow placebo-controlled randomized trials to go forward. We'd be more able to understand the merits of challenge trials. So Near al. Has gone into this in detail. Challenge trials are tests of vaccines in which a vaccinated person is intentionally infected. They're also called controlled human infection trials. To go against the well-known and not too accurate summary of the Hippocratic Oath to do no harm, but these participants are highly informed about the risks. They know what they're doing, and they're choosing to participate in these trials. The World Health Organization agrees that challenge trials can be done ethically, and even challenge trials with SARS-CoV-2. These 8 criteria here are pretty sensible and pretty straightforward. I think with challenge trials in particular, site selection is important. A biohazard unit is probably needed. Participant selection needs to be done very carefully. And of course, the final criterion, informed consent, is critical. It has to be extremely rigorous. Challenge trials can save lives. Professor Andrew Lowe, who will be speaking a bit later, did an analysis with colleagues showing how challenge trials under certain conditions, easily met, can save lives. Now, in some cases, other designs might be more helpful. But regardless, we need to be prepared so that we can move quickly to set up vaccine trials of any kind. And as Near al., as all of us have noted, we have to be prepared well in advance. And if we are, then the challenge trials can be very low risk. One idea is to create a volunteer vaccine corps, kind of like the Peace Corps. This could be a group of volunteers standing ready for any kind of vaccine trial, but it would be particularly helpful for challenge trials where the risk to any individual might seem to be a bit greater than in a conventional placebo-controlled randomized clinical trial. As we've heard, one day sooner is a group that is gathering volunteers for challenge trials, and they already have several tens of thousands. So the trolley problem yet again. So we have the vaccine trial trolley problem involving volunteers in a kind of vaccine volunteer corps. It's a purely active choice with a full knowledge that some in the volunteer corps could be harmed. They are pulling the switch themselves, putting themselves in harm's way. knowing that a couple of them might even die in order to have a chance of saving many more lives. They're willing to do this. Now, as Nir Eyal notes, of course, we don't know which one of them is going to be harmed, and it might be none of them, but they're the ones who are actually taking the choice themselves. So they are actually pulling the switch. They have complete control over this choice, which makes it perfectly ethical. Now, Preston had the great idea that we might be able to solve a lot of problems at once by combining three innovations: powerful deep immune profiling that Nir Eyal has described, and then secondly, a pan-coronavirus vaccine, and third, what we're calling a step-up challenge trial that a broad-spectrum vaccine would enable. Deep immune profiling makes possible many things, not just better vaccine trials. There's a lot of clinical utility, not just advantages for research. We can get a clearer idea and earlier of what differentiates mild from severe disease, which can greatly help fine-tune treatment choices, for example. And the correlates of protection can yield very important information about whether or not vaccines in the general public have been successful. And then, of course, stratification of degree of protection that helps us safely select subjects for challenge trials. So in sum, under the assumption or hypothesis that our PanCoV vaccine works to some degree against all human coronaviruses by targeting highly conserved portions of the viral family, challenges could start with a less pathogenic coronavirus, like human coronavirus OC43. Then immune profiling data would be collected for determining correlates of protection by the vaccine. After this initial low-risk phase, other participants would be challenged with a more serious pathogen, so it's SARS-CoV-2 with a coronavirus study. These other participants would be selected from a totally separate group, also vaccinated with PanCoV, who have a post-vaccination immune profile similar to those in the first group, who are most protected against the milder coronavirus by the same vaccine. And then, of course, all the subjects will be followed long-term to ensure safety and to see how protection changes over time. There's a lot more detail here, of course, that you can read in our new clinical trial white paper, which should be on our website very soon. Just go to radvac.org and click on white papers. I don't think it's up there quite yet, but it will be soon. So there are a number of benefits to the RaDVaC step-up trial with the PanCoV vaccine. It's a relatively safe way to conduct challenge trials, we believe. Much less expensive than conventional vaccine trials. It's not cheap, but it's still much less expensive. Success could mean at least some protection in the population against new coronaviruses, not just SARS-CoV-2. That's in fact the point, or one of the points. The vaccine can easily be modified and retrialed to target more specifically any new coronavirus, and the infrastructure can be repurposed for non-coronavirus families. And it might even help prevent some types of the common cold. So I want to close by strapping on my clinical trials liaison hat a little bit more tightly and putting away my trolleyologist hat, and note a few things about why it could be valuable to create the infrastructure needed to produce and test via step-up challenge trial a broad spectrum vaccine like PanCoV. It's a free and open source platform for vaccine development, testing, and production. The vaccine is broad spectrum, very easy to produce, and it should permit relatively affordable, unique trial designs that quickly yield results, and we think safely. It's highly scalable. The ingredients are safe, and the intranasal delivery means there'll be less vaccine hesitancy, which is actually really important. I think a big part of vaccine hesitancy actually is not crazy theories about the CIA putting in a tracking chip, but it's actually just a fear of needles. So that means broader vaccination coverage, which means we have a greater chance of reaching herd immunity or reaching it sooner. The presumed induced mucosal immunity, as Jutta Paulus has pointed out, may significantly reduce transmission. The design permits rapid updating to cover emerging variants of concern. Full local control is possible. Vaccine testing and clinical trials infrastructure can be used to address any current or future pandemic. And we have to remember, we have a huge, important current crisis that we have to solve. But ultimately, we at RaDVaC, and I think many of us here, are interested not just in open source vaccines, but the power of open source science in general. So vaccine testing and clinical trials infrastructure can be the catalyst for broader health sector and biotech development in various regions and around the world. But beyond that, each separate group working on this can become part of a global open science network, which need not be globalized. And that's what we'd really love to see created and would love to help create with everyone here. So let's create the Linux of vaccines and open science. Please get in touch if you have ideas about how to help move this forward. We'd love to hear from you. Thank you very much.